All right, thank you so much for inviting me. First and foremost, very happy to be here. I'm the CEO of 28 Bio. So what we're basically doing, in a nutshell, if you will, is engineering human brains at scale. We're building human brains, so-called human brain organoids, in the lab with engineered cognition. I'll talk about in a little bit what that means. We are replicating key features of the human brain structure and connectivity. And what I'll show you is that our models exhibit complex real-time brain activity patterns. In addition to that, we have the ability to stimulate and record the human brain organoids, which gives us the ability to train and develop learning and memory capabilities. All right, I think the problem statement doesn't have to be sort of stated in this audience, but I'll do it anyways. 92% of neurological drugs fail when tested in humans. One of the highest failure rates we have in our healthcare and life science system and R&D. Yet, as an industry, we still spend $28 billion trying to develop neurological drugs. We keep failing, and we keep getting surprised when they fail. One of the main reasons for that is the fact that the preclinical predictions simply don't work, right? We've cured a lot of neurological diseases, Alzheimer's, Parkinson's, so on and so forth, in mice and animals. We just haven't done it in humans, right? And the problem is this translatability and the difficulty it is to predict which drugs are actually going to work in humans when they actually eventually go into the clinic. So our Nexon platform screens preclinical drugs for efficacy and toxicity to replace ineffective animal models. We do it both on healthy cells to identify toxic effect or disease cells. I'll talk a little bit about in a few slides on what disease cells, to identify therapeutic rescue or converting disease into healthy. Ultimately, what we measure is EPHIS data, right? Electrophysiological data. And we measure features that are very similar to the actual EPHIS features that are measured in the clinic. Very, very close collaboration with Dr. Hartung here at Johns Hopkins. For those of you that are familiar with his research, he's really pioneering the field of organoid intelligence. Organoid intelligence is the development of brain organoids and their ability to learn, remember, and interact through biocomputing systems. And part of our IP is in-licensed from Hopkins and Hartung's lab. So one key component in this technology is this proprietary neural interface technology that allows us to stimulate and record what's happening to the brain, which means that we can now actually reinforce behaviors, adaptive behaviors, related to memory and learning. First off, obviously, we got to grow and cultivate an organoid or a tissue system that represents the key neurological cells needed. Here are some examples of those cells: neurons, astrocytes, etc. We also grow oligodendrocytes if we want to mimic and model out myelination, for example, microglia to incorporate immune capabilities. But the goal, the North Star really, is to build a system that, to the greatest extent possible, replicates the capabilities of parts of our human brain. So what we can do is then train organoids to remember how to respond, right? So again, the ability to stimulate and record gives us the ability to literally shock the organoids and see how they respond. We can give them a very harsh shock and almost kill them. We can give them a very balmy stimuli that they like more. And that allows us to reinforce a predictive response. So you see here, we have a training phase where we constantly shock and see a different response in different time bins. And then we have a resting phase. And then when we shock or stimulate the organoids with the same pattern, we get the same response, meaning they have learned something, right? And the theory behind this is that if we then train them over time, they learn, they develop memory capabilities that allows them to do certain things with the information that they have just learned or remembered. And then eventually they will lose that memory capability and they will forget the information. That was the theory, the thesis to the left. We actually conducted a set of experiments where we proved that out, that we can actually learn and train them over time. They will remember the behavior and then eventually forget it. We asked ourselves, okay, how do we actually measure this? How do you prove that this is the case? So we trained them to play Pac-Man. So this is basically an exercise, an experiment, where we had trained them to run away from the ghost and move towards the fruit. And what we saw over time is that the longer we trained, the better they scored, right? And now you can obviously bring this capability into a drug development setting. We can develop completely novel biomarkers for neurodegenerative diseases in regards to memory and learning. All right, Takeda, one of our customers, has published a study that shows how our platform, the Nexon platform, compares against the traditional animal model. They screened 84 drugs with known clinical neurotoxic outcomes. And the study shows that the Nexon platform, the brain organoids, are 7.4 times more predictive of clinical outcomes than animal models. And this is really a core kind of foundation of the predictability proof, if you will, of the platform's ability to predict human outcomes in drug development. We launched a little bit over a year ago, and we already have a number of pharma companies on the platform: Pfizer, Merck, Takeda, Roche, Sanofi, GSK, AZ, Janssen. These are all drug development applications, either toxicity or efficacy. We've now added the US Air Force, and we just added the US Army for their defensive weapons programs, where we test different nerve agents for neurotoxic effects. And then also, as of about a month ago, FDA is also a customer of ours. You might have seen a number of announcements that came out very recently where the new administration, FDA administration, NIH administration too, basically announced that they're going to phase out animal models. They're expensive, they're ineffective, and we're killing a lot of animals at the same time. The NIH came out a few weeks ago and said, from now on, we are not going to fund any NIH grants that only have animal models. You need to include NAM data, so-called alternative models to animal models, such as our platform, to be eligible. Customers are buying more and more. Here are 3 key accounts that we're working with since launch. As you can see, Takeda, Sanofi, and Genentech. Orders are picking up over time as they get more and more adoption inside of these organizations. FDA, I mentioned, is one of our customers. FDA is actually screening a drug that's been on the market for some time that has shown neurotoxic data. And the FDA has told us if the model reads out positive, meaning the compound is neurotoxic, they will withdraw it from the market. So it tells you a little bit about how this transition is happening now within the FDA and the NIH as well in regards to adoption of non-animal-based technologies. In addition to the screening business, we also have used the data coming from a screening business to train an AI model to really try to elucidate the fundamental biology underlying some of the diseases we screen drugs for. It's a novel target discovery platform that we then partner with pharma on. The service business, some of the customers I mentioned, is about a $5 billion total TAM. The partnership business, which is the target discovery, and then pharma partnership from the AI modeling work, is about a $15 billion TAM in neurology. We're not the only ones going after this, but we are the company that has the highest throughput. Throughput is incredibly important when it comes to servicing pharma with drug screening capabilities. And we have basically gotten to 384 wells on a plate. Each well is literally a microbrain, if you will, or a micro organoid. Myself, to the left, I'm joined by Dave Wiener. He's a neurologist in drug development as our CSO, actually developer of Neplasia and safinamide. Also joined by Stu Gibb as our VP of Sales and Marketing and Commercial. You may be familiar with another organ chip company, Emulate, that's a liver on a chip. Stu took Emulate from 0 to $50 million in revenue before he joined us. All right, we are close to break even. We're going to reach break even second half of next year. We launched last year. So pretty significant growth curve. And then we're going to scale beyond that. Right now, the focus is on the screening business. When we reach break even, we're going to put more focus into the AI discovery commercialization engine and business. We are raising right now $6.5 million. 4.3 is already taken, so we got 2.2 left. The two goals for this raise: first goal is to reach EBITDA positive second half of next year, and then expand the data lake for our AI target discovery business. All right, thank you.